So we talked about gram-positive cocci. A couple weeks ago, I drew something up here kind of showing you gram-positive cocci, gram-negative cocci. It gets complicated when we get to gram-negative bacilli because there are so many groups. So we've already talked about fastidious organisms that are gram-negative bacilli or gram-negative coccobacilli. Right? That's our Haemophilus, our Francisella. What are some clues that lead us to thinking that our organism is a fastidious organism? Doesn't grow on sheep blood agar. What about if it's a gram-negative bacilli, what kind of agar should it grow on? Yes, but there's a selective agar. MacConkey. We expect gram-negative bacilli to grow on MacConkey. But a big clue that it's in this fastidious group for gram-negative bacilli is no growth on MacConkey. There's another group, non-fermenters. This is speaking to glucose, not lactose. So if it's unable to ferment glucose, it's called a non-fermenter. And we'll talk about those next week. Let's see, there's curved, I think curved, like your Vibrio and your Campylobacter. They're curved gram-negative rods. And I might include them in with like your miscellaneous oddball gram-negative bacilli. But the biggest group is your Enterobacteriaceae. Enterobacteriaceae. And this is a family of gram-negative bacilli that are commonly found in your intestinal tract, in soil, in those kind of places. And there's a few characteristics that we'll talk about that all Enterobacteriaceae share, and that way we can, hopefully we can categorize our organisms into that group, and it'll help us narrow down what our possible pathogens are. Because how we identify organisms is different based on which category we're in. I want to say there was something else, but I can't remember off the top of my head. So, this is MacConkey agar, and they took some E. coli, and since E. coli is a lactose fermenter, it grows pink. So it's E. coli on MacConkey agar. So these are your objectives. You should be able to list the general characteristics of organisms that belong to this family, Enterobacteriaceae. So we'll talk about a few characteristics that all of the organisms, for the most part, share. State the name of the gram-negative bacilli associated to specified diseases. What kind of diseases or infections do they cause? Describe various methods used to identify gram-negative bacilli. And we talked about a lot of different biochemical tests back in Chapter 9. We talked about TSI, citrate, urease, motility. We're going to be talking about all those kind of tests again. And then describe the pathogenesis of clinically relevant members of the family Enterobacteriaceae. So just focus on the organisms that we talk about in this presentation. If I don't talk about it and you see it in your book, don't worry about knowing it. Okay, so these are the most common clinically relevant organisms that cause disease in in humans. So how do they cause disease? Give the organism's characteristic growth on non-selective agar, selective and differential media, and then presumptively identify the isolate to the genus level. So based on how they appear on sheep blood agar and MacConkey agar, or HE agar, XLD agar, based on their color and their appearance on those types of agars, we can at least get to a genus level to identify it. And then, given the key reactions for identification, be able to identify your organism to the species level. And then finally, develop an algorithm using biochemical tests to identify the clinically significant Enterobacteriaceae. So we'll learn a couple tricks of the trade to how to easily identify gram-negative bacilli in this family. So here's the case in point. We've got a 69-year-old diabetic woman who's hospitalized for an amputation. She complains of chest pain and a persistent cough and shortness of breath. So they took a sputum sample and plated it on routine media. So that's your sheep blood, chocolate, MacConkey agar probably. After 18 hours of incubation, the MacConkey plate showed moderate growth, a 3+ growth, of an oxidase-negative, non-lactose fermenting organism. Sheep blood agar plate showed very moist, large mucoid colonies. The biochemical tests to identify the isolate were performed, and these were the following results. The TSI slant was acid over acid with gas. The MVIC reaction, which we'll talk about shortly, it's just an acronym for 4 different tests. It was negative, negative, positive, positive. Urea was hydrolyzed, so it was urease positive. Lysine decarboxylated and malinate utilized, and the organism was non-modal. So based on those biochemical reactions, hopefully in the future, once you study, you'll be able to identify this organism. So what's the significance of this patient's health status and medical history? She's kind of at risk for opportunistic infections, right? She's diabetic, so her immune system, she's at risk for wound infections. Her immune system isn't up to par. She's in the hospital. She's had something amputated. She's probably in bed for a long period of time. So respiratory infection is likely. So she's got a lot of things at risk. And then the colony morphology provides clue about the identity of the organism. Let's see, we've got growth on a MacConkey plate. What does that tell us? It's a gram-negative bacilli. It's non-lactose fermenting. So what color is it on MacConkey? Anybody remember? Colorless, right. We just saw E. coli in the very first slide. E. coli was pink on MacConkey because it ferments lactose. So if it doesn't ferment lactose, it'll be a clear colony. So the fact that it's clear on MacConkey leads us down a certain route for identifying our organism. And then what biochemical tests are most helpful? You'd probably go insane if you memorized every single biochemical test for each one of these organisms. So if we can clue in on some very unique reactions, it can help us identify our organisms. The fact that this organism is non-modal is a big clue. Okay, so the general characteristics for Enterobacteriaceae, or another word is enterics. They all ferment glucose. Okay, they all reduce nitrate to nitrites, so they all reduce nitrate, all nitrate positive. They're all oxidase negative. Of course, there's an exception. Plesiomonas is an exception, but we're not going to talk about Plesiomonas. Most of them are motile. Okay, so the fact that an organism is not motile is very significant. Okay, Klebsiella, Shigella, and Yersinia are the only 3 that are not motile. And Yersinia is motile only at room temperature, so that's a big clue if we see that in a test question. So Enterobacteriaceae, or enterics, or another word you might hear is coliforms. You think of your colon. They all reside in the GI tract. So their presence in other organ systems is not common. Colony morphology for Enterobacteriaceae, they're gram-negative coccobacilli, or straight rods. And their colony morphology on sheep blood agar is usually not very helpful. All your gram-negative bacilli, or all of your Enterobacteriaceae, are basically large, flat, and gray. Mucoid, some of them are mucoid. They're facultative anaerobic, so they'll grow throughout the entire tube on a thioglocolate broth. They'll grow in an aerobic atmosphere and an anaerobic atmosphere. They're macroscopic morphologies, what I just touched on, large, gray, moist colonies. Some of them can be described as mucoid, very, very moist looking. So if we see the word mucoid, we want to think of certain organisms. And probably the most common one, if you want to link mucoid and Klebsiella. This guy was sometimes I've seen him so mucoid that if you hold a plate up like this, he just drips down. It's disgusting. So growth on media. Selective and differential agar is very helpful for us to distinguish between the different gram-negative bacilli. So MacConkey is both selective and differential. It inhibits gram-positive organisms. So if you have growth on it, you can assume that it's a gram-negative bacilli. And then it's differential based on lactose fermentation. So if it does ferment the lactose in the media, and there's only lactose in MacConkey agar, there's no glucose, then the colonies will appear pink. If it's unable to ferment the lactose, then the colonies will be clear. EMB agar, or eosin methylene blue agar, is also selective and differential. It inhibits gram-positive organisms, and so gram-negative bacilli are the only ones that grow on it. Its lactose and sucrose fermentation allows it to be differential. So it's got 2 sugars in it this time. And so organisms will appear certain colors based on whether or not they're able to ferment those sugars. HE agar, again, is another selective and differential media, and this is very helpful to very quickly. It's mostly used to recognize Salmonella and Shigella. So it uses bile salts to inhibit gram-positive organisms and some of your gram-negative organisms that are routinely found in the intestinal tract. So what your goal for this type of media is to isolate your Salmonella and Shigella. Well, these guys don't ferment sugars, the lactose and the sucrose in the media. Salmonella and Shigella are non-fermenters. They don't ferment those sugars. So they'll appear clear. Whereas most of your non-pathogens are able to ferment the sugars in the media, so they'll appear a certain kind of color. So if they ferment lactose and sucrose, they'll appear an orange color. Whereas if they don't ferment, they're described as having a blue-green color, but they're actually colorless because the agar itself is blue-green. That makes sense? So you'll see when we look at a non-lactose fermenter on MacConkey agar, the MacConkey agar is pink. So the colonies almost take on the appearance of the pink agar underneath them. But the pink colonies from a lactose fermenter is very different than the color of the media. So you might see that in a test question. You might see blue-green color, or blue-green color with a black center, or you might see colorless. They're the same thing, it's just the colonies appear like the media. So HE agar is green. Here's an example of the MacConkey. Here's a lactose fermenter on MacConkey agar, and notice we've got the growth of the colonies here where they're nice and pink, right? And then also notice that there's this pink haze diffusing out from the growth. That's called precipitated bile. Notice this one doesn't have that pink haze growth or pink haze diffusing away from it. This one does. So this is pink colonies, very mucoid, right? This is a non-lactose fermenter on one side and lactose fermenter on the other side. Here's the HE agar. See how the agar's green? So these are they call them blue-green colonies, but they're actually clear colonies, but they look like they're blue-green because of the color of the media. So here we've got normal flora on this plate, and we've got some kind of a pathogen, right? A possible pathogen. Salmonella and Shigella are supposed to be, well, Shigella should be clear, like this, but Salmonella is clear with black centers. So this is an example of Salmonella. It's clear, and then we've got black centers. Here we've got some clear colonies with black centers. So if we saw that on a stool culture, we would have to rule out the possibility of this being Salmonella. All right. And then what about these orange colonies over here? What do we know about that? What does the appearance of orange colonies tell us? If our pathogens are non-fermenters, our non-pathogens are fermenters, right? So if they ferment the sugars in the agar, they're going to turn this orange, yellowish orange color. So I wouldn't do anything to any kind of colony that's an orange or yellow color. I don't care about those. They're supposed to be there in stool. But if it's colorless or if it's clear with black centers, then I've got some work to do. And it's easy to open that plate and look at it and notice that there's something there that requires my attention without having to sift through all of the junk that's in a stool culture. All right, here's another one, XLD agar. And different facilities will use different selective and differential media based on whatever they choose to use. So XLD agar, again, is selective and differential. It inhibits gram-positive organisms and lets your gram-negatives grow. It can inhibit some of your usual gram-negative organisms. This one's got 3 carbohydrates, sucrose, lactose, and xylose, and it's got a phenol red indicator. And it's also got some thiosulfate in there so that we can detect H2S. So here, again, it's basically the same kind of thing as the HE agar. We're looking for Salmonella or Shigella. We're looking for the pathogens, which are non-fermenters. And so they are going to appear colorless, whereas our non-pathogens are fermenters, and they're going to appear yellow because they'll ferment the sugars in the media. So what we're looking for are colorless colonies or colorless colonies with black centers. And so XLD agar is red to begin with. So you might see in test questions that you're looking for red colonies or colorless colonies. So keep that in mind. I got a picture here. See how it's nice and red media? Clear colonies, but this is an H2S positive organism, so the centers have turned black. What about this one? Should we work that organism up? Is it a possible pathogen? Is it fermenting the sugars on this agar? No. So it's a possible pathogen. Could be Salmonella, could be Shigella. So we would have to do some more testing to rule that out. What about this one? Is that a pathogen or a non-pathogen? Non-pathogen because it's not colorless. It's clear, or it's yellow, excuse me. What about this one? Notice we've got 2 different colony types. We've got these little ones here that have no color, and then we have these ones that have some black in the center. They're both pathogens, right? They're both possible pathogens. So one might be Shigella, or one might be Salmonella, because Salmonella is H2S positive. It has black centers, whereas Shigella does not. Okay, so that's the colony morphology on certain types of media can really help us to identify and spot certain pathogens in the Enterobacteriaceae family. The virulence and antigenic factors, I think your book goes into depth about some of these. I'm not going to go into depth. I'm going to focus on a few concepts here. Adherence, to prevent being removed from tissue. If you think about your urinary tract system, the flow of urine through the genital tract actually washes out the genital tract, right? they're not able to, if they can't hold on to those endothelial cells, if they're washed out, then they can never set up infection, right? They've got to hold on to that tissue and then start replicating and growing out of control in order to start the infection. So if they have adherence properties, they might have like little pili or little hooks, if you will, that can latch onto those endothelial cells and allow them to penetrate the tissue and start replicating. Okay, if they can't grab on, if they can't adhere to the tissue, then they can't set up infection. So toxins like food poisonings, they might produce spores, or they might produce some kind of secreted toxin that can cause disease. Invasive enzymes or spreading factors, they might produce an enzyme that breaks down fat or breaks down some kind of a immunoglobulin protease like IgA. It might break down those proteins and allow it to cause disease better. Antibiotic resistance, we're not going to talk about this in depth, but extended spectrum beta-lactamases are getting more and more common in infections. And then you might have heard carbapenem-resistant Enterobacteriaceae in the news lately. CBEs, or CRE, excuse me. You haven't heard about it? So we're seeing some increased resistance in your Enterobacteriaceae, and very difficult infections to treat. Serology, there are different types of antigens that can be located on the cells, and I'm not going to go into depth about each one, but I wanted to just point out to you the O antigen and the H antigen, because we'll talk about E. coli 0157H7. And basically what that is it's characterizing a certain strain of E. coli based on the antigen that's present on the surface of that cell. So this one in particular has the antigen 0157 and the antigen H7, and that's a pathogen. So the clinical significance for Enterobacteriaceae, usually they're normal flora in your gastrointestinal tract. So if they cause infections, it's usually of two different types. Majority of them are opportunistic pathogens. What does that mean, opportunistic pathogen? Maybe the immune system's depressed. Yeah, there's a lot of things that might lead to an opportunity to set up infection, right? If you're on broad spectrum antibiotics and a lot of your normal flora is killed off, that normal flora that is present could grow unchecked, right? Your normal flora helps protect your body and keeps things in check. But if all of it's killed off, then other things can grow out and cause infections. Or maybe if you've got a scrape in your skin, right? That's an opportunity for something to cross that skin barrier, right? So an opportunistic pathogens are usually considered normal flora, but they cause infections when they are given the opportunity. And there's tons of different ways that they can see an opportunity. Let's see, if they get into a non-normal site. So your gastrointestinal, these are Enterobacteriaceae, they're supposed to be in the GI tract. What if they get in the urinary tract system? They're not supposed to be there, right? So they could cause infection. E. coli is normal flora in the stool. But if it gets in the urinary tract system, you're going to have a urinary tract infection. It's not supposed to be there. Or what else? If you have a big gaping wound, if the E. coli gets into that big gaping wound, it's not supposed to be there. And so it's taking advantage of an opportunity. Let's see, also hospitalization, certain medical procedures, you're at risk. Catheters, you're at risk for an infection from an organism that doesn't usually cause infection. Primary pathogens are organisms that cause disease no matter what. They're not supposed to be there. So when they are there, they're considered a pathogen and they'll cause disease. So your Salmonella is not normal flora in the stool, and so if it's there, it's going to cause a problem, right? Shigella, same thing. It's not supposed to be in your stool. Yersinia pestis causes the plague, right? So it's not normal flora. If it's present, it's going to cause infection. There's also Yersinia enterocolitica, and that's an organism that causes diarrhea. So if it's present in contaminated food and water, that's an opportunity, right? Okay, so this is just kind of spelling out some different Enterobacteriaceae and the common diseases that they cause. So we'll talk about, looks like we'll talk about most of these. E. coli, it can cause, number one disease that E. coli causes is urinary tract infection, but it can cause other kinds of diseases as well. Shigella primarily causes dysentery and diarrhea, but a lot of these you'll see repetition, like bacteria, bacteria in the urinary tract, pneumonia, septicemia, opportunistic infections, wound infections. So you see a lot of duplication there. And some of them cause a very specific type of infection. So Yersinia pestis causes plague. There's no other disease that it causes. It causes bubonic plague. Okay, so knowing that, we're going to start talking about the different Enterobacteriaceae, the different characteristics that they have, and infections that they cause. So E. coli's, he's the big dog in this group of organisms. They are dry and pink colonies on McConkey plates. So here's a good picture of E. coli, and we've got the big zone of precipitated bile. It's described as dry colonies, and we'll in a second. Let's see if we got any moist. Oops. In a little bit, we'll see an example of very moist colonies. On EMB agar, E. coli grows this metallic sheen. It ferments sugars very quickly, and so it's other organisms will appear slightly different if they ferment sugars. E. coli is the only one that causes, it's like a fish scale kind of sheen, green sheen on EMB agar. It's very often beta hemolytic on sheep blood agar, not always, but many times. And they're just described as large, flat, and gray on sheep blood. Very strong lactose fermenter. So the biochemical reactions that we see for E. coli, of course they ferment glucose. Every organism in this group ferments glucose. But it also ferments lactose, trehalose, xylose. Those aren't really important. I want you to know the lactose. Something else it does is it's indole positive. So if we have tryptophanase, if we have tryptophan in the media, then E. coli will break it down into indole. So it's indole positive. Glucose fermentation is through the mixed acid pathway. So we talked about the MR and VP test. So it's MR positive, VP negative, and then it's citrate negative. So these are the reactions. Here we've got IMVIC here. That's an easy way for us to remember several biochemical reactions for an organism. So it stands for indole, methyl red, Vogus Proskauer, and citrate. So E. coli is positive, positive, negative, negative. Indole positive, MR positive, VP negative, citrate negative. So if we can remember that E. coli is IMVIC, positive, positive, negative, negative, then we've got 4 reactions that we know. Some clinical types of E. coli. Your book goes into a lot of depth into the different types of E. coli that are present. I'm not gonna spend a lot of time on this. Uropathogenic E. coli. This is the #1 type that we see, and it causes UTIs in humans. Most common cause of UTIs in humans? That's a good test question, right? It's got pili on there that allow it to grab onto the epithelium or the tissue in the GI tract, and then it also produces some cytolysins to help it evade phagocytosis. Now, there's also some GI pathogens for E. coli, right? There's 5 major types. Let's see. Traveler's diarrhea. E. coli causes traveler's diarrhea. It can cause dysentery. Enterohemorrhagic E. coli is where we see a lot of blood in the diarrhea, and that's caused by E. coli 0157H7. Has everyone heard about that at some point? And then there's others too. So just be aware that if you see that kind of terminology, I'm not going to make you memorize that kind of stuff. But I did want you to know about E. coli 0157H7 because it's a pathogen that we have to rule out when we do a stool culture. And there's lots of different ways that you might do it. Okay, there's not just one way to rule it out. So this particular strain of E. coli produces toxins called the Shigella toxin. So we can do immunoassays to detect that toxin. We can do a serology to detect an antibody against E. coli 0157. And we can see if the titer increases. A 4-fold increase in the titer indicates a recent infection. You can also do screening of stool with sorbitol-containing MacConkey. So instead of lactose in your MacConkey agar, you have sorbitol. And so E. coli 0157H7 does not ferment sorbitol, whereas the normal E. coli does. So here's a picture of both types on MacConkey agar. And so this pink colony is your normal E. coli, whereas this colorless colony is your pathogen, E. coli 0157H7. So it allows us to spot it really quickly, really easily. Of course, we'd have to do some testing to confirm that, but it allows us to spot it very quickly. Okay? All right, the next group we're going to talk about is your Klebsiella, Enterobacter, and Serratia. And this is the KISS group, okay? I think about the band KISS, the KISS group. And if we group them together and realize or know the common characteristics of the organisms in this group, hopefully we can memorize things a little bit more easily. So they share a lot of properties. Again, they're common opportunistic and nosocomial infections. What's a nosocomial infection? An infection that you get in the hospital. Good. So its biochemical reactions, I remember it's opposite from E. coli. So its MVIC is negative, negative, positive, positive. Okay? And one easy way to remember that, notice Enterobacter is very similar to E. coli. The name. They both start with E. For some reason, I always got E. coli and Enterobacter confused. So I try to remember that Enterobacter is opposite from E. coli. So if I can remember that E. coli is positive, positive, negative, negative, then I can remember that the KISS group is opposite. Okay? So let's see. I thought I had... So these are the common characteristics. These are the biochemical reactions that each of these 3 organisms share. And now we're going to talk about each organism in particular and the biochemical reactions that we use to differentiate them from each other. So Kleb pneumo is your most common isolate in this group, in this genus of Klebsiella. We see a lot of it in lower respiratory tract infections and urinary tract infections. It's gray on sheep blood agar, large and gray, but a lot of times it's described as mucoid. That's that runny, snotty looking one. So here's a picture of it. It looks very shiny and very wet on MacConkey agar. It's non-modal. Very few Enterobacteriaceae are non-modal. So the fact, if we see non-modal in a test question, we want to think of this organism along with 2 other organisms. It's urease positive, so it creates ammonia byproduct, which is very stinky, pungent odor. It's a sucrose fermenter, so that's going to help us in a bit. VP positive, okay, and virulence factor. This guy can produce a capsule, which can help it evade phagocytosis. And so many times if they produce a capsule, they're going to be very mucoid. Let's see. And this guy is one of the leading carbapenemase-resistant Enterobacteriaceae that you'll hear about. Its counterpart is Klebsiella oxytoca. So it's very similar to all these other reactions that Klebpneumo has, but there's one key biochemical reaction that differentiates it from Klebpneumo, and that's that it is indole positive. Okay, Klebpneumo is indole negative. Remember our IMVIC reactions? What were our IMVIC reactions for this group? Right, so indole negative for Klebsiella, but this guy's a little different. He is indole positive. Okay. Now, Enterobacter species, the most common isolates are Enterobacter cloacia and Enterobacter aerogenes. The infections that cause wounds, urine, blood, spinal fluid, biochemical reactions that are important. Enterobacter cloacia is urease positive, so it's going to be very pungent, stinky odor. Enterobacter cloacia is going to be lysine decarboxylase negative, whereas aerogenes is positive. So that helps us differentiate between the two. I prefer to use the urease because it's easier to remember. There's both sucrose fermenters, and they're VP positive. Now, Serratia marcescens, we saw him close to the beginning of the semester. He is unique in the fact that he can be red, have a red pigment. So here he is on McConkey agar. He's not a lactose fermenter, but he appears red on McConkey. It's very different color, if you remember, than a lactose fermenter. And then you can kind of see it in here on the chocolate agar. So if you see red pigment, you want to think of Serratia marcescens. And if you think Mars is the red planet, you can remember red. Very highly resistant to antimicrobials. It causes urinary tract, respiratory tract, bacteremia, just like all the others. So your biochemical reactions, it's ONPG positive. That's pretty unique. So it's a slow lactose fermenter. DNA is positive, gelatin positive, and VP positive. Now, Proteus. Proteus mirabilis and Proteus vulgaris are the most commonly isolated species. Probably mirabilis is the more so than the other. Again, isolated wounds, ears, blood, urine. One common, very unique colony morphology that we see is that it swarms. And it doesn't always swarm, but when it does, we want to think of Proteus. So here, if we took a loop, if we touched a colony and took with our loop and touched it in the center of this plate and then didn't streak it out, just stuck it in the incubator, the Proteus will swarm out. See the concentric circles? It just takes over the entire plate. It'll grow over everything. So it's very difficult to isolate a colony because this Proteus will grow over everything that's on the plate. So you have to use selective agar to isolate your Proteus when it swarms. Another colony morphology characteristic that's very unique to Proteus is the burnt chocolate odor that you smell. We do have one in lab this week, a Proteus, and it is ripe. Usually I smell the burnt chocolate odor, but this one is just awful ripe. It's PDA positive, phenylalanine deaminase positive. So if we see that, that's very unique. I always think PDA at the prom, and that helps me remember that Proteus, Providencia, and Morganella are PDA positive. you're only going to have to remember Proteus. I'm not going to ask you to remember Providencia or Morganella. So remember, PDA at the prom, Proteus. It's a non-lactose fermenter, so that characteristic helps us to identify our organism because there's not many that are non-lactose fermenters. It's urease positive, very pungent odor. H2S positive, that helps us a lot when we're narrowing down our identification. And then gelatin positive, that helps us too. So how do we tell the difference? If we have a Proteus, how do we differentiate from Proteus mirabilis and Proteus vulgaris? Well, Proteus mirabilis is indole negative, and it does not ferment sucrose. So it'll be K over A on H2S, and H2S positive on TSI agar, whereas Proteus vulgaris will be acid over acid because it ferments the sucrose in the TSI slant. And then vulgaris is indole positive. What's a positive indole reaction? What's it look like? Do you remember? Red. It is red. So a positive, and we'll do tons of indoles in there, so you'll remember after a while. So I think of vulgarity and red. I link those. So that helps me remember that my Proteus vulgaris is red. It's indole positive, and my mirabilis is not vulgar. Okay. Okay, Citrobacter. Citrobacter frondii is the most common isolate. Let's see, biochemical reactions. It's urease positive, very stinky. Ferments lactose. It's citrate positive, and that's easy to remember because Citrobacter, right? Citrate, Citrobacter. It's MR positive, and it's H2S positive. This guy is the only lactose fermenter that is H2S positive. So if I see those two reactions together, I know it's a Citrobacter. Okay, so primary intestinal pathogens of the family Enterobacteriaceae. So those were all opportunistic pathogens. Now we're going to talk about our primary pathogens. These guys cause infection no matter what. So Salmonella, you want to think clear, colorless colonies, non-lactose fermenters with black centers. So Salmonella does not ferment sugars, but it's H2S positive. So in your HE media or your XLD media that detects hydrogen sulfide production, he'll be colorless, but he'll have a black center. Biochemical reactions, notice is negative for all of these, but it's H2S positive. That's a big thing there. Clinical infections, this is kind of easy. If we've probably heard of Salmonella. What does it cause? Diarrhea, dysentery, yeah. Most of our gastroenteritis organisms will cause diarrhea. You might have some vomiting or abdominal cramps. It's caused by, let's see, 10 to the 6 organisms. So it's caused by the organism itself, not a toxin that it creates. Like E. coli O157H7 produces a toxin that causes the infection, whereas the organism causes the infection, not a toxin. Okay? And it causes 10 to the 6 organisms. How many is that? 1, 2, 3, 4, 5, 6. So you have to ingest this many organisms in order to get an infection. It's quite a lot, huh? Unless your potato salad's been sitting out all day. So it can, we see it in peanut butter, milk, eggs, pet handling. Pets can get it, poultry, cereal, dog food, tomatoes. It can develop into a carrier state. So the person may not actually have diarrhea. They may not have the symptoms of Salmonella, but they're still carrying it, and they can still pass it to other people. You've heard of Typhoid Mary? She had Salmonella typhi, so she caused a lot of problems because she kept going into the food industry and spreading Salmonella to all the patrons of the restaurant. She just served it up with all the meals. But she carried Salmonella in her gallbladder, and she spread it to all these people. And then there's also non-typhoidal bacteremia, let's see, and then some other enteric fevers. So there's different species of Salmonella. We're not going to learn how to speciate them, but just kind of keep it in mind. Salmonella typhi is the one that causes the typhoid fever. So think of typhoid Mary. Now Shigella, I remember Shigella as the do-nothing organism. He's non-motile, no urease production, no H2S on TSI, no decarboxylation. He's a non-lactose fermenter. He does nothing. Okay? There's different species of Shigella. So if you do isolate a Shigella genus from your stool culture, there are some latex agglutination tests out there. So you just mix a little of the colony with each of the antibodies. So it'll be a kit with 4 different antibodies. An anti-group A antibody, if it agglutinates, if your colony agglutinates with the group A antibody, then you know you have Shigella dysenteriae. Or if it agglutinates with Salmonella Species antibody, you know you have that species. So some of these have some antigens on their surface that allow us to use those kind of methods to speciate them. So it causes dysentery, so we can see local inflammation, fever, chills, a shedding of the intestinal linings, mucus, blood, ulcer formations, rectal prolapse. Shigella dysenteriae is the most severe infection that we see out of the 4 different types. It's usually non-fatal. It's just kind of self-limiting in developed countries, but in countries where there's poor health care and poor sanitary control, it can be fatal. It's got a low inoculum. Notice here, 10 to 200 organisms can cause disease as compared to Salmonella, which was 10 million. Only need a very, very few Shigella to cause your infection. It's spread through the fecal-oral route or by flies, so your 4Fs will cause this GI tract infection. And we can see it in certain populations like daycare centers, crowded populations. Children that are under 10 and infants are very susceptible to this. They can pass away very quickly because of the violent diarrhea. Yersinia species. This is the last of the primary pathogens that we'll talk about. So you've probably heard of Yersinia pestis that causes plague. Bubonic systematic. I can't say it. Someone say it for me. Septicemic, there we go, and pneumonic forms. We're not going to really learn the different forms of plague, but keep in mind there's different presentations and forms of the disease. It's spread through the bite of infected fleas. Gram-negative coccobacilli. It has a safety pin appearance, so it'll stain more intensely on the ends than it will in the center of the organism. So if you see these characteristics, you want to think of Yersinia. Preferential growth at room temperature, so it grows good at room temperature, and it's only motile at room temperature. Usually when we do a motility test, we incubate it at 37 degrees, which is body temperature, but it's not motile at 37 degrees. It's only motile at room temperature. So if you see that, you want to think of Yersinia. And we can see this. It can contaminate platelets because our platelets are incubated at room temperature, whereas our blood supply is kept at colder temperatures. Yersinia enterocolitica is the other species in this genus, contaminated food and fecal material. And this refrigeration is ineffective for this group. It can survive in very cold temperatures, so it can live in our blood supply and reproduce. And then acute gastroenteritis and appendicitis-like symptoms. So we do have a particular type of media that we use to spot Yersinia, to grow it. It's called CIN agar. And Yersinia appears like a clear colony with a red a red bullseye. So here it is. CIN should be on your media worksheet. So make sure that you link CIN, auger, and this colony morphology with Yersinia. Okay, so laboratory diagnosis. How do we diagnose Enterobacteriaceae? For specimen collection and transport, usually it just depends on the presentation of the patient. If they've got a wound, then you're going to collect, hopefully an aspirate is the best sample, probably a swab. So the same kind of specimen collection and transport applies. There's no special handling. It just depends on the presentation of the patient. For isolation and identification, let's see, the direct microscopic exam or the gram stain isn't helpful to differentiate between these organisms. For staph and strep, we saw chains and clusters, so that helped us kind of differentiate. Or we saw lancet-shaped if it was strep pneumo, and that helped us differentiate between them. But these are all gram-negative bacilli, so that it doesn't help us here. When we culture them, they grow very quickly, very easily. Identification, let's see, there should all be oxidase negative. You want to perform your oxidase off sheep blood agar. You don't want to do an oxidase off selective and differential media. Same as for your indole and your catalase and those kind of things. It should ferment glucose and it should reduce nitrate. So if it has these characteristics, then you know that you're dealing with an Enterobacteriaceae. Okay? So you're going to do a certain battery of biochemical tests to identify it completely. Let's see, screening stool cultures for pathogens. There's a lot of normal flora in the stool, so our biggest help is that selective and differential media that we use, so we can spot those clear, colorless colonies. And then you can also use serological grouping, like your monoclonal antibodies to the antigens on the surface can help you identify. And there's some available for both Salmonella and Shigella. So here's just an example of some of the organisms, the Enterobacteriaceae. So it starts out with TSI agar. The TSI agar is a test that we do to determine if it ferments glucose, right? So K over K means it does not ferment glucose. So we would go to a non-different panel. We would use non-lactose or non-glucose fermenting methods to identify. Here we've got K over A, H2S, acid over acid, K over A. Notice all of these, they're glucose fermenters. They may or may not ferment lactose or sucrose. So let's see. You can memorize that if you want to. Here's a helpful chart from your book. You'll notice when you open your textbook, you've got pages and pages of your organisms in alphabetical order, and then you've got 2 pages of biochemical tests, and then it tells you if it's 99 here, then that means that 99% of the isolates are positive for that test. Okay, so if you can go to that chart and see if your Citrobacter is motile or not, we're gonna learn how to identify organisms a little bit differently than memorizing all of these. Okay, and there's your case in point. So we're going to look at a video